Learning outcomes. After studying this module, you shall be able to know what is blood and the composition of blood. You will be able to examine the blood. You will be able to know the source of blood. How will you know that examined sample is of human or animal? That also you can examine. You will be able to know what is the age of blood stain. You will be able to identify the blood groups and confirmatory test for blood. Introduction. Blood. Blood is the red fluid that circulates in our blood vessels, that is veins and arteries. The main function of blood is to act as the body's transport system, but it also has a major role in the body's defense against infection. There is no substitute for blood. It cannot be made or manufactured. Donors are the only source of blood for patients who need it. Blood is one of the most important physical evidence which is frequently encountered at the crime scene as a pool of blood, droplet, stains, etc. It can be found in almost every type of criminal activity involving physical violence like murder, assault, rape, etc. at crime scene in the form of valuable evidence. Now, blood stains are of two types. One is the visible blood stain and the second is the invisible blood stain. Now, let us learn about the components of blood. In vertebrates, it is composed of cells suspended in blood plasma. Plasma, which constitutes 55% of body blood fluid, is mostly water, that is 92% by volume, and contains dissipated proteins, glucose, mineral ions, hormones, carbon dioxide, that is the plasma being the main medium for excretory product transportation, and blood cells themselves. Albumin is the main protein in plasma and it functions to regulate the colloidal osmotic pressure of the blood. Now, first is the RBC, which is also called as erythrocytes. Red blood cells, also called erythrocytes, are the most common type of blood cell and the vertebrate organism's principal means of delivering oxygen to the body tissue via blood flow through the circulatory system. RBCs take up the oxygen in the lungs or gills and release it into tissues while squeezing through the body's capillaries. In humans, mature red blood cells are flexible and oval biconcave disc. They lack a cell nucleus and most organelles in order to accumulate maximum space for hemoglobin. Approximately 2.4 million new erythrocytes are produced per second in human adults. The cells develop in the bone marrow and circulate for about 100 to 120 days in the body before their components are recycled by macrophages. Each circulation takes about 20 seconds. Approximately a quarter of cells in the human body are red blood cells. Next component is WBC, which are also called leukocytes. They are the cells of immune system that are involved in defense system of the body, that is protecting body against various infection, diseases and foreign invaders. All leukocytes are produced and derived from multipotent cells in the bone marrow known as hematopoietic stem cell. Leukocytes are found throughout the body including the blood and lymphatic system. 5 different and diverse types of leukocytes exist. These types are distinguished by their physical and functional characteristics. Monocytes and neutrophils are phagocytic in nature. Next is platelets, that is also called thrombocytes. These are the component of blood whose function along with the coagulation factor is to stop bleeding by clumping and clogging blood vessel injuries. Platelets have no cell nucleus. They are fragments of cytoplasm which are derived from megakaryocytes of the bone marrow and then enter the circulation. These unactivated platelets are biconvex discoid, that is lens shaped structures, 2 to 3 micrometer in the greatest diameter. And the last component is the plasma. Blood plasma is the pale yellow liquid component of blood and normally holds the blood cells in the whole blood in suspension. This makes plasma the extracellular matrix of the blood cell. It makes up about 55% of the body's total blood volume. It is the intravascular fluid part of extravascular fluid, that is all body fluid outside the cell. Now dear student, we will learn about the major functions of the blood, that is the transportation, maintenance of body temperature, control of pH either acidic or basic, removal of toxin from the body and the excretion. Now let us learn about the examination of blood in detail. First is the physical examination. In the natural light, the blood stain appears as brown, reddish brown, clot or crystals of reddish brown color. If the stain is clear and visible, then examine it under UV light at 230 or 269 nanometer of wavelength. Next is the presumptive or screening test. The blood stain obtained from suspected area contaminated with the material should be tested for positive blood stain. Presumptive tests produce a color, reaction or release of light in presence of catalytic property of the blood. Now next is the phenolphthalein test. It is also known to be Kastel Meyer test. Phenolphthalein is reduced by zinc powder in a strongly alkaline medium. If this reduced phenolphthalein is oxidized by oxygen liberated by the action of peroxidase on hydrogen peroxide, that is H2O2, then a pink or purple color is obtained. If the stain of blood, the sensitivity of this phenolphthalein test is about 1:5 lakhs. The reagent which are used, first we have to make stock solution in which 2 gram phenolphthalein, 20 gram potassium hydroxide, 100 ml distilled water, 20 gram zinc dust. Next we have to make working solution, ethanol, that is 10 ml, phenolphthalein stock, that is 2 ml, distilled water 10 ml, ethanol 2 ml and hydrogen peroxide 10 ml. Now reagent preparation. The reagent is formed by adding phenolphthalein 2 gram, potassium hydroxide 20 gram, distilled water 100 ml. Mix and add zinc powder and boil under reflux, that is 2 to 3 hours, until the stock solution is formed. Cool and decant into a bottle containing some zinc to keep in the reduced form. Now add ethanol, that is 10 ml and phenolphthalein stock solution 2 ml, distilled water 10 ml and again ethanol 2 ml and 3% hydrogen peroxide H2O2, that is 10 ml as working solution. Hydrogen peroxide is used in every color reaction, or in other words, it is responsible for color obtained in the reaction. If the color obtained is pink, then it confirms the presence of blood. Now let us go through the procedure. A small cutting swab or extract of the suspected blood stain is placed on a filter paper or spot test paper. 2 or 3 drops of ethanol are placed on the stain. 2 drops of working phenolphthalein solution are added to stain. After waiting to ensure that no color develops at this stage, 2 or 3 drops of hydrogen peroxide are added. An intense pink color indicates the positive test for peroxidized activity and indicates the presence of hemoglobin. Next is tetramethyl benzidine, that is TMB test. In this we use the reagent acetate buffer, that is sodium acetate 5 gram, glacial acetic acid 13 ml, distilled water 57 ml. Then we also make working solution in which we use TMB solution 1.5 gram, acetate buffer 20 ml. Now let us go through the reagent preparation. First we have to take 1.5 gram of benzidine and 13 ml of glacial acetic acid. Then we will add 57 ml of distilled water. After shaking, benzidine solution is ready for test. Now let us go through procedure. Place cutting or swabbing of the stain on a filter paper or spot test paper. A drop of TMB solution is placed on the stain, followed by a drop of 3% of hydrogen peroxide and mix with the glass rod. Appearance of immediate blue-green color is a positive test for peroxidized activity, that is indicative of presence of hemoglobin. Next is luminol test. Luminol reagent we have to make first, in which we take sodium perborate 7 gram, 3-aminophenol hydrazide 1 gram, sodium bicarbonate 5 grams. Then we prepare the reagent. The reagent are formed by taking 0.7 gram of sodium perborate and 1 gram of 3-aminophenol hydrate is mixed with 5 gram of sodium bicarbonate. Now comes to the procedure. Take the suspected blood stain and add few drops of luminol reagent. The appearance of fluorescent color indicates positive test of the blood. Next is Leucomalachite Green test. Reagent Leucomalachite Green 1 gram, sodium perborate 3.2 gram, glacial acetic acid 65% solution, that is 66 ml, distilled water 33 ml. Now reagent preparation. The reagent of Leucomalachite Green test is formed by the combination of Leucomalachite Green, that is 0.1 gram, sodium perborate 3.2 gram and glacial acetic acid 66 ml and 33 ml of distilled water. If the color appears to be green, it confirms the presence of blood. Now let us go through the procedure. Add Leucomalachite Green solution on the stain. If green color appears, it indicates the presence of blood. Now next are the confirmatory tests of blood. First is Takayama test. It is also known as hemochromatin test. Takayama reagent, saturated solution of glucose, that is 3 ml, pyridine solution, that is 3 ml, 10% of NaOH, that is sodium hydroxide solution 3 ml, glacial acetic acid 7 ml. Reagent preparation. Take 3 ml of saturated solution of glucose, 3 ml of pyridine solution and 3 ml of NaOH solution along with 7 ml of glacial acetic acid. Now come to the procedure. Place the material to be tested on a microscopic slide and cover with a cover slip. Add a drop of Takayama reagent and allow to flow under the cover slip. Warm the slide gently on a hot plate at 65 degrees Celsius for about 10 to 20 seconds. Allow to cool and observe under microscope at 100x. The appearance of pink needle shaped crystal of pyridine hemochromatin, that is pyridine ferroprotoporphyrin, is positive reaction for heme and confirms the presence of hemoglobin. Now next is Teichmann's test. First we have to prepare Teichmann's reagent, in which KCl, KBr, KI is used 0.1 gram each, then glacial acetic acid 10 ml. Now reagent preparation. For the Teichmann test, the reagent is formed by combination of KCl, KBr and KI at about 0.1 gram each in 100 ml of glacial acetic acid. The reagent reacts with hemoglobin and gives brownish rhombic crystal which confirms the presence of blood. Now let us come to the procedure. Place material to be tested on a microscopic slide and cover with a cover slip. Let the reagent flow under cover slip. Warm the slide gently on a hot plate at about 65 degrees Celsius for 10 to 20 seconds. Allow it to cool and observe under microscope at 100x. The appearance of brown rhombohedron shaped structures of crystals ferroprotoporphyrin chloride is a positive reaction for heme. Next is the microscopic test. Microscopic method of examination is having paramount importance relating to the identification of blood stain. First is determination of the species of origin of nucleated RBCs along with the cell structure. Next is the sex determination by examining chromatin bodies in the leukocytes. Then the detection of blood related pathological condition. Next is spectroscopic method. In this method, identification of hemoglobin and its derivatives is done by characteristic absorption band when viewed through microscope. Now let us go through the test. A small portion of suspected stain as small as 2 mm is put in 0.5% of potassium cyanide solution for 15 minutes and then filtered. The filtrate thus obtained is taken in 1 cm. Cell and UV light is passed with spectrometer. The absorption is observed in 300 to 600 micron. The absorption maximum is obtained at 422 millimicron, shows the presence of cyclohemoglobin. Next is detection of species origin of blood. The biological evidence has been identified necessarily to determine and confirm whether it is of human origin or not. If it is non-human origin, then to which species it belongs, to the species specific protein in the blood stain or other body tissue may be identified with the help of species specific antibodies. The species specific protein for blood stain or tissue are extracted in normal saline, that is 8.5 gram sodium chloride in 1 liter of distilled water or 5% of ammonia solution. The various methods are applied to detect the species origin. Like the first is Precipitin tube method. Take 6 Precipitin tubes, number can vary on the number of antisera used, and place them vertically in a Precipitin tube stand and label them. Put a drop of the blood stain or tissue extract in the tubes. Carefully add 1 drop of antiserum for species origin, that is anti-human serum, anti-fowl serum, anti-dog serum, anti-cow serum, anti-goat serum, etc. along with the walls of tube. Leave the undisturbed for 30 minutes at room temperature. Then Carefully examine the white ring at the interference of two solutions. If precipitate is formed, it belongs to specific antiserum. Now next is the double diffusion method. In this method, both the reactants, the antigen and antibody, diffuse towards each other in agar gel plate, and when a antigen combines with its specific antibody at optimum proportion, precipitin are formed. Fill the central well with tissues extract and peripheral wells with different antisera for species origin like anti-human serum, anti-fowl serum, anti-dog serum, anti-cow serum, anti-goat serum, etc. Cover the petri dish and keep gel in a moist chamber for overnight. Now examine the gel for the presence of precipitin band formation. Next is the crossover electrophoresis. In a buffered gel, the strain extract antigen is placed in cathodic well and the antiserum in the anodic well. When electric current is applied, the globulin antibodies migrate cathodically because of electroendosmosis. The other serum protein migrate anodically. A precipitin reaction occurs midway between the paired well when an antigen combines with its specific antibody. Fill the cathodic side well of the pair with stain extract and anodic well with species specific antiserum. Again, it may be of anti-human serum, anti-dog serum, anti-cow serum, anti-goat serum, etc. Now, place the slide on the electrophoresis chamber. The stain extract should be nearest to the cathode and antiserum nearest to the anode, and connect the gel to tank buffer chambers by two pieces of filter papers on each side. Run electrophoresis for 20 minutes at 150 volts. Record the conditions. After the electrophoresis, switch off the power supply and remove the slide, and then observe the slide with aid of a lamp. A fine white line of precipitin between a pair of wells is a positive reaction. Now next is the age of blood stain. Age of blood stain can be calculated by color and nature of the stain. Fresh blood stains are bright red in color, moist and sticky. Within 24 hours, the stain turns reddish brown in color. After 24 hours, the stain turns dark brown and finally black. Next is detection of blood group. After determining the origin of blood group is to be determined. It is also to be examined by prosecution to mention the blood group. The modern serological techniques have derived blood in 2 or 3 constituent classes for discrimination of human blood, that is AB, AB and O. The blood group of typing antigen. Although several types of antigens have been discovered so far, but in case of dried blood stains are concerned only with ABO, MN and RH antigenic system have been found of practical utility in crime work. Next is the polymorphic enzyme. These enzymes are located in red blood cells which are responsible to catalyze biochemical reaction in the body. The enzyme which have same functions to perform but different physico-chemical properties are called isoenzymes. Many isoenzymes have been identified in dried blood stain using electrophoresis techniques. Few examples of these isoenzymes and phosphoglucomutase PGM and adenylate kinase AK, erythrocyte acid phosphatase EAP and esters D. PGM system which have 3 common variants PGM1, PGM21, PGM2 has been found very stable. Next is the RH system. This system, however, now not found useful to analysis of dried blood stain. It is more often used in disputed paternity cases in which whole blood analysis is done. Next is the factors affecting detection of blood group from blood stain, like putrefaction and thermal condition. Now let us summarize what we have learned in this module. Blood is an important evidence found at the crime scene that leads to the further investigation of various crimes. Blood examination has an important role in solving suicidal, accidental and homicidal cases. Various tests are performed to identify and confirm blood stain of human or animal origin. Identification of paternity and maternity cases by blood grouping. Species and origin of blood detection gives indication of various animals. And the last is the determination of age of the blood stain. Thank you.